Good morning, I am Madhurima Datta, Zoology and Biology educational content creator. Now's content for B.Sc and M.Sc in Zoology Honours, Microbiology Honours, Physiology Honours, Biochemistry Honours and Biotechnology. And M.Sc also in these subjects. An exam for CUET PG exam, GATE exam and CSI NET Life Sciences exam. Paper Evolution, Unit 1, Evidences for Evolution. Today I will discuss about the analogous organ. What is the analogous organ? Analogous organs are biological structures across different species that perform similar function but differ fundamentally in their anatomical constructions, embryonic origin and evolutionary ancestry. Next, core concept and mechanisms, that is homoplasy and convergent evolution. Analogous structures are the primary morphology evidences of the convergent evolution. Unrelated or distantly related organisms independently adapt to similar environmental pressure or ecological niches. It is leading to functional convergence without a shared common ancestor for the trait. Next mechanism is lack of common ancestry. Unlike homologous organs, analogous structures do not derive from a common ancestral anatomical structures. Mechanism is genomic and developmental perspective. Developmentally, these structures are governed by distinct gene networks, that is embryonic tissue layers and developmental pathway. Example, insect wings derive from the thoracic epidermal outgrowths, whereas bird wings derive from the modified vertebrate forelimbs. Next, classic example. At first, in animal kingdom, wings of insect versus birds and bats. Insect wings, it is chitinous membranes supported by veins, non-osteological cuticular growth, and bird wings, bony forelimbs covered with feathers. Bat wings, bony forelimbs stretched with cutaneous patagium membrane. Next, eyes of cephalopods versus vertebrates. Both features are lens, retina and cornea. Cornea is a camera type eye. And differences, cephalopod eyes are eversely wired and photoreceptor face forward, no blind spot. Whereas vertebrate eyes are inversely wired, that is nerve fibers pass through the retina, creating a blind spot. Next example is fins of fish versus flipper of dolphins or whales. Hydrodynamic adaptations for propulsions in aquatic environments. Fish fins are supported by fin rays, that is pterygophores, and dolphins flippers are modified, that pentadactyl tetrapod forelimbs with hyperphalangy. Next example in plant kingdom, stem tendrils, that is grapes versus leaf tendrils, that is peas. Both serve for climbing support, but grape tendrils derive from modified stem axis, and while pea tendrils derive from the modified leaflets. Next, thorns, that is citrus stem modification versus prickle, that is rose cortical epidermal outgrowth versus spines, that is cactus modified leaf. All serve mechanical defense against herbivores, that is despite divergent anatomical origins. Next, modern evolutionary concept in analogous organ. First is deep homology versus analogy. Advances in evolutionary development biology, that is evo-devo, and reveal the seemingly analogous structures are sometimes regulated by homologous genetic toolkits. For instance, the PAC6 gene, that is the master, regulates eye development across both invertebrates, that is Drosophila, and vertebrates, even though the adult eye structures themselves evolved independently as analogies. And next point is phylogenetic reconstruction. Analogous character represent homoplasys, because in cladistic analysis, homoplastic traits can introduce false signals, that is parsimony errors, if misidentified as shared derived characters, that is synapomorphism. And advanced phylogenetic algorithm use maximum likelihood or Bayesian inference alongside molecular data to filter out convergence artifacts. Next, evolutionary mechanics and selection pressure in analogous organ. Analogous organ arises through convergent evolution driven by common adaptive landscape. First is selection on functional constraints. Environmental regimes impose physics-based constraint on body plans. For instance, fluid dynamics dictates that high-speed underwater locomotion requires a fusiform body shape and hydrofoils. As a result, sharks, that is chondrichthyes fish, and ichthyosaurs, that is reptile animals, and dolphins, that is mammalia, independently evolved streamlined bodies, and dorsal fins and flappers are present in their body. Next, amorphology and ecological niches filling. Unrelated taxa occupying identical ecological niches in geographically isolated regions often develop analogous trait. Example, that is North America placental wolves versus extinct Tasmanian marsupial wolves, that is thylacines, showing near identical skull mechanics and dental adaptations. Next, evolutionary trade-offs and developmental constraints. Analogous structures illustrate how evolution is constrained by existing body plans. First is exoskeleton versus endoskeleton constraints. Arthropods cannot scale up analogous structure, that is respiratory trachea or jointed appendages, to match vertebrate dimension due to the weight of a chitinous exoskeleton and oxygen diffusion limits. Next, analogy via expectation. Structure often evolve function independently by co-opting existing traits for new uses. For example, the thumb of the giant panda is not a digit but an enlarged radial sesamoid bone co-opted to strip bamboo leaves, and analogous counterpart to the true opposable thumb of the primates. Now the topic has ended. For all video of B.Sc and M.Sc in zoology, botany, microbiology, physiology, biochemistry and biotechnology, please subscribe my YouTube channel Biogenesis by Madhurima and follow my Facebook account Madhurima Datta. Thank you everyone.